Thank you. It's a pleasure to be here. Thank you very much for inviting me to come and talk about some of the things that we've been doing in Europe. It's a pleasure to be here. I think that I'm just a few kilometers away from the exact opposite of the other side of the world where I'm coming from, because I'm from Spain. And currently I am I'm establishing a remote sensing laboratory at the University of Melbourne, focused on aspects related to precision agriculture, biotic and abiotic stress detection using technology, like imaging spectroscopy, hyperspectral remote sensing, thermal imaging, using both manned and also unmanned vehicles with a laboratory for calibration purposes and trying to derive physically based remote sensing techniques and approaches that allow us to map large areas of both crops and vegetation, natural vegetation, in the context of stress, as I said before. In the last, we've been working in the last year or so on getting this University of Melbourne Airborne Imaging Spectroscopy facility that we are now flying with different instruments that I will talk about a bit later in my talk, because some of them are quite innovative in aspects related to imaging spectroscopy and hyperspectral imaging. In the last around 5 years before I arrived in Australia, I've been working detached from my government position in Spain in the Research Council as a senior scientist working for the European Commission, particularly one of the institutes, which is in northern Italy. And the main focus was to develop new methodologies and algorithms for the early detection of two harmful and big problems we had in Europe, and we still have them, which is the Xylella fastidiosa and the pinebot nematode. The only way we could handle this big problem that happened in Europe, particularly arriving in the year 2013 officially to Italy, but then spreading from there to other countries like the Balearic Islands that belong to Spain, but also France, and going into other countries as well, is that we had to develop and to explore innovative technology, innovative methods, because of the virulence and the big spread of the Xylella. That's just in a couple of years. That's the picture in Gallipoli, one of the areas in southern Italy, and that what happened just a couple of years later. It's been a massive spread, and standard remote sensing or other surveillance methods were not optimum for the detection of the disease. The problem got the attention, big attention internationally, both in the media but also in the scientific journals, wondering if the olive trees could be saved in southern Italy. But it's not only the olive trees, because because of the Xylella can be spread into more than 500 different species, as you know perfectly. After this big impact in the media and in the international scientific journals, the European Commission launched different funding strategies, and we got involved in European projects focused entirely on the Xylella fastidiosa, with more than 29 partners around Europe, with from more than 14 countries, including some of them that were not part of Europe. The successful eradication of Xylella, it was a consensus internationally that had to deal with two different issues. One of them is that it was very critical to have an early diagnosis. Secondly, that a small infected area was required. That was not the case of southern Italy, but that was the case of other places in Europe, particularly in Spain or in the Balearic Islands. And a very critical aspect is that the development of fast methods for the early detection of the disease across large areas is absolutely critical. I think it was very much highlighted in all venues that scientists were meeting that we really have to develop fast methods for the large-scale early detection of diseases across large areas using innovative methods. I don't think we are talking about assessing or monitoring severe or moderate, even low level of symptoms. We are actually trying to detect at the earlier stages trying to differentiate between asymptomatic and symptomatic initial levels of the disease because of the severity, because it's at that stage where we can really make changes and we can really detect it, trying to avoid the spread. The standard remote sensing methods that are normally used can detect the low, moderate, or severe levels quite easily because of some of the plant traits and some of the indicators that we can get from remote sensing are heavily affected by or heavily affected by the defoliation and the fact that leaves get chlorotic or actually they fall down. But in the case of the asymptomatic versus initial symptoms, which is that case, we can hardly see it in the field, and in that case we had to develop methodologies based on the physiology and linking with innovative remote sensing aspects. The fact is that in this particular disease and other ones in perennial crops, it takes almost more than a year sometimes to develop the symptoms, and during that time it's very important to detect it because otherwise the host can be acting as a source of inoculum for other trees around. So the innovative aspect really came from the fact that we had to look for new technology in remote sensing, linking with knowledge that we had already in physiology, because some of the indicators that we are trying to look at are really small, subtle, very minor changes that happen in the physiology of the plants at the earlier stages. When our eyes cannot see it, the plant is already affected, and small changes in the absorption due to different photosynthetic pigments and non-photosynthetic biochemical constituents that we have in the plants are what we are trying to detect by using a remote sensing method using specific cameras. Secondly, innovative indicators that international scientific community it's developing for quantifying photosynthesis, and in fact NASA or the European Space Agency is planning the launch of satellites for the global coverage of and global monitoring of chlorophyll fluorescence. Those are also indicators quite innovative that normally are not used in the remote sensing arena, but are very useful for the early detection of diseases because of a very tiny part of the re-emission of light from vegetation can be detected from sensors, and that's directly linked with photosynthesis. And the photosynthetic changes that happen to infected vegetation at the earlier stages can be a very important indicator of the early previsual stress. As the plant gets more stressed, chlorophyll fluorescence decreases, and that decrease in chlorophyll fluorescence is one of the many screening indicators that we could detect or we could use as a fingerprint for the detection of the disease. Finally, also canopy temperature, because of the reduction of transpiration that plants under stress undergo, and that decrease in the evaporative cooling that is not happening when the plant is not transpiring because of the disease can be also detected with high-resolution thermal imaging that we could fly both on board drones or manned aircraft. The fact is that the typical standard well-known, widely used remote sensing couldn't be used in this case because of that is used usually based on either monochrome or a small number of spectral bands. Is what we are calling normally in remote sensing multispectral remote sensing, that we are recording spectral bands, discrete spectral bands at different wavelengths. We really wanted to had to go to something else, which is imaging spectroscopy, something that is called hyperspectral imaging, which means that for every single pixel in our images, we are actually collecting the full hyperspectral, full signatures of the radiation that is reflected by the plants, which are directly related to the physiological processes happening behind. Instead of a number of discrete spectral bands, as we can see here, we are actually collecting the entire electromagnetic spectrum that allow us to derive signatures which are related to particular diseases. I think I went too fast. Yeah. Okay, so here we can see the concept of imaging spectroscopy and scanning the surface with hyperspectral images that as the plane or the satellite or the drone is flying, we are collecting the full electromagnetic spectrum, and that allows us to get full scenes of the hyperspectral images. That's from a natural vegetation. That one is from vegetation and crops, where we can then extract the full spectrum from every single tree. In the case of orchards, every single tree will have the signature, and that signature is what is really analyzed in order to look for specific plant traits which are related to the early detection of the particular disease. This is happening because of the very large push in technology that happened in the last 20 years. That's one of the first hyperspectral imagers that, in fact, I was operating that one back in Canada in the year 2000. In just about 10 years, that's the evolution of the sensors. That's my hand, and that's also a hyperspectral imager that, instead of a massive, more than $500,000 instrument, we are now talking, and also very, very heavy, that could be only flown over on board a very large plane, very expensive. We are now talking about less than half a kilo in just about 10 years, or around 300 grams or so in 2015. That's also my hand. That's also one of the instruments in my lab, or even getting much, much smaller. So, I mean, the revolution in technology really happened in the last less than 20 years in imaging spectroscopy, which allow us to fly and to use these sensors in any possible platform that we can think of, not only manned aircraft, but also unmanned vehicles, or any mobile platform that it's available. So we have tested, we've been testing in the last around 5 years this technology and the methodologies. I mean, the innovative aspects of technology linked with remote sensing, hyperspectral imaging spectroscopy, high-resolution thermal imaging, and physiology-based remote sensing. I mean, trying to understand, not just to analyze the signal as a signal, but trying to understand the physiological processes behind the changes that we detect using remote sensing. And we did it in the context of the Xylella fastidiosa outbreak in Europe. So we've been working for 2 consecutive years in the Puglia region, that's southern Italy, collecting thousands of hectares with thousands of trees being evaluated in the field by plant pathologists. But also in the newest outbreaks that happen in Europe later on, which is the case of the Alicante, which is in the east part of the mainland Spain, and also in the Balearic Islands in the Mediterranean Sea, where the whole island is infected by Xylella fastidiosa, affecting not only olive trees, but also almonds and other species. Field data would allow us to get the ground truthing of the different plant traits that we were evaluating in the context of early detection of the disease with different instruments, spectral-based instruments that evaluated the optical properties and the changes, subtle, small changes in the optical properties of the vegetation undergoing, in this particular case, Xylella fastidiosa infection, and conducting airborne flights from southern Italy and also from Spain to evaluate with hyperspectral, thermal, multispectral, RGB, CIR cameras, all the possible plant traits that we could detect using remote sensing methods. That's the typical high-resolution images that we can collect with airborne imaging spectroscopy, where we can identify every single tree over large areas. Large, I mean in this case, thousands of hectares in one single flight. And in the bottom right, you can see the full spectrum in the visible and the near-infrared with hundreds of different spectral bands. The typical would be to collect 3, 4, 5 spectral bands. In that case, we're talking about more than 250 spectral bands collected for every single pixel of every single image, and extracting the pure radiation reflected by every single tree, and then applying different algorithms. And the same thing with the thermal data. That's a mosaic obtained with thermal imaging, and we can, applying algorithms, detect the within crown variability and within orchard variability due to the presence of the disease, because of the direct link between thermography, between canopy temperature, and the stress induced by Xylella fastidiosa. The results of the project got quite good attention internationally, talking about flying cameras to spot the disease because of, in Europe, as you perfectly know, it's a really big problem, Xylella fastidiosa, but also in scientific journals that where we published the results by looking at the spectral plant trait alterations due to the objective of the previsual symptom detection of Xylella fastidiosa. We could detect different things. We could detect which are the most important plant traits that are linked with a particular disease, with a particular Xylella fastidiosa. Not all screening traits are affected equally depending on the diseases, and that's something that we are now evaluating in the context of other diseases that are happening and where we have databases from. But the fact is that some of the standard indicators, like NDVI, anyone in remote sensing knows what NDVI is. It's a normalized ratio between two spectral bands that 90% of the people working remote sensing would use. Well, we could see that they were not good indicators of the early detection of stress, and that we had to use more advanced remote sensing approaches based on other indicators which are totally impossible to get from standard remote sensing. We could identify the specific plant traits which are affected by Xylella fastidiosa. We could study the robustness from one year to another, and even we are now doing is to assess whether these plant traits can be applied from one country to another, for example, from the Italian outbreak into the Spanish databases that we have collected. The robustness across years is really important to see, because all the screening methods, all the screening parameters that we can retrieve from hyperspectral remote sensing show that from year to year are quite robust, which makes more robust models for the early detection of the disease. The results that we got are that depending on the methodologies, depending on the type of inputs that we used from different remote sensing sensors, but also the modeling approaches using deep learning or more linear discriminant analysis approaches, we show that we can get up to around 83, 82% of overall accuracies in the early detection of Xylella fastidiosa by using hyperspectral imaging. If we didn't use hyperspectral imaging, then we will go to less than 60, around 65% or so. That is much lower. But the positive aspect of the 80-something% overall accuracies, which was good news in the context of harmful disease like this one, we were wondering about what is the remaining 20% or 18%. I mean, why are we making, it's remote sensing, the one making the errors here, or what is exactly happening? So what we did was to revisit the false positives. False positives were those that remote sensing was classifying as infected, but the plant pathologists in the field were saying that they were healthy. So we decided to go back and after the first flight and field evaluation, we revisited half of the database, because that was part of the project in which we were monitoring all the fields affected in that southern Italian region. And we revisited around more than 1,700 trees. And, but then out of those, we focused particularly on the false positives. We wanted to know what happens to the false positives that remote sensing said that were infected by Xylella fastidiosa. The results showed that the probability of a tree, of a false positive tree getting infected was always higher for the false positives than for the true negatives. What that means is that remote sensing was telling about trees that apparently they were not infected, and the plant pathologists couldn't capture that in the field, but remote sensing was actually with months in advance detecting those trees as infected, and that's why they got showing symptoms in every revisit that we got. So because of the probability, showing that the probability was higher. We are now dealing with trying to develop hybrid models that combine both physical and artificial intelligence approaches, in which we are trying to solve some of the difficulties in the operational use of physical models that require, in some cases, several inputs and are quite difficult to operate, and they are quite difficult to use in the context of model inversion and operational remote sensing data collection and analysis of the data sets. Some final conclusions. I think that we feel that the early detection of Xylella and other harmful diseases at the pre-visual stage is possible using innovative remote sensing methods. We are now also checking into verticillium wilt, Phytophthora, pinewood nematode, I mean, all these other important diseases that where we have databases from and that have been collected using hyperspectral remote sensing. How much of what we have learned from Xylella can be applied? What is the distinct signature of the plant traits that we get from Xylella as compared to other fingerprints that can be distinct for other diseases? I think that's quite interesting to assess. I think that everyone in this room would agree that it's critical to detect the Xylella or other harmful diseases-induced symptoms at the early stage when infected areas are small. But I have to add that if that is the main objective, we should forget in some way about the standard remote sensing methods that we are normally using, because the standard remote sensing methods will not be useful for the early detection of the diseases. It will be useful for mapping areas affected. And in fact, when I was in the European Commission, the European Parliament asked how many trees have been killed, how many trees have been affected. Okay, yeah, sure, we can map millions of trees using a standard remote sensing methods, but if the objective is the early detection, we cannot do it with the typical standard remote sensing approaches. Adoption of new operational remote sensing methods is needed for surveillance purposes, so that's linked with the previous one. We need to go to advanced, innovative technology in remote sensing that use spectral regions, spectral bands, and retrieve plant traits that cannot be detected using other standard remote sensing approaches. I think that collaboration has been critical in this particular case of Europe. I guess that, well, I mean, the situation in Europe is a bit different in the sense that we are forced to collaborate all together. We cannot get funding from the European Commission if we are not going together from different countries at the same time. But in this particular case, the multidisciplinary collaboration has been critical because it's not just a remote sensing problem, it's not just a plant pathology problem, it's not epidemiology problem. I think it's a problem of all of us working together, adding different experiences and knowledge. And finally, I really think that innovation is absolutely critical. I have to say, I was in some way at the European Commission, as part of this project, the one deciding which technologies we had to use or which ones we wanted to explore. Standard remote sensing methods would have failed totally if that was the decision that had been made based on the previous experiences. Standard remote sensing methods would have been useful to map how many trees were affected and how many trees were killed, but we wouldn't have made any progress on the actual early detection of diseases and that particular Xylella. Technology currently provided by vendors or service providers would have failed, nothing against, but the most innovative aspects of remote sensing are currently coming from research institutions and universities worldwide. I think it's critical to link with industry and to transfer the know-how from university and research institutes to industry, but the actual and the current vendors and service providers are not really offering the latest in innovation in the aspect of remote sensing. And finally, I think this is quite critical, innovation means risk. I think that risk needs to be funded. If we don't fund risk and if we don't fund new ideas and innovative ideas that might work but may not work, perhaps, I think that we are not really getting the most of everyone that is around working on this aspect. So I think that it's really important for funding agencies, and I would like to encourage funding agencies to look a bit less on the risk involved and probably look more into the potential outcomes that we get from it. And that will be all. Thank you.